introduce our next speaker then, Dr. Kevin Bewley from the UK Health Security Agency. SARS-CoV infection in lower human respiratory MPS at high containment as a platform to support pan-betacoronavirus vaccine evaluation.
That's a terrible title. Who came up with that?
Thanks for that.
So the way we make vaccines at the moment for SARS-CoV-2 is we look at what's hanging around sort of in May, and then we make a guess on what's going to be hanging around in November. We make a vaccine to that, and sometimes the booster is good, sometimes it's not so good. So there's an aspiration to make a vaccine that will boost all of your beta coronavirus activity to protect you regardless of what happens to be evolving as the variant of the day. And a broader aim is that we can make a vaccine that will protect against those coronaviruses we haven't yet seen jump across into the human population. Let's see what this slide looks like. So we're working with CEPI. They work with vaccine manufacturers, and we put those through our animal models, and we see protection against maybe SARS-CoV-2 and SARS. We also want to include MERS. MERS is a problem because there isn't really a very good animal model for that unless your facility can hold camels or llamas. So one way around that we're attempting is we're creating human alveolus on a chip, and that's what this thing here is. We populate that with primary human cells. We've got airway cells on one side of a membrane, we've got endothelial cells on another side, and we recirculate the media underneath. And at the moment, we're right at the beginning of this project, so we are exposing these human airways to virus and just characterizing what that infection looks like. So we're looking for virus shedding kinetics, we're looking at cytokine expression, and trying to show that that is a model of what you would see in a natural human infection. And what the hope is we can take the sera from our animal models and we can put them into the human model and support the fact that, say, we see protection in a hamster against SARS-CoV-2, that it will also protect a human alveolar cell in the context of the chip. So I haven't got, I think actually I have. I have got a little bit of data. So two of the measures that we've measured so far, we're looking at the barrier function. So the barrier breaks down and your alveolus becomes more leaky. So that's something we're particularly worried about. And also looking at shedding of SARS-CoV. So we have shown already that we've got sustained shedding of virus from the apical surface, and also some evidence that the virus is making it into the endothelial compartment, which is the equivalent of breaking out into your bloodstream. Very transient, but it looked like it was there. There are other readouts that will come on stream. This is basically a current experiment. So the poster's only a half-done thing at the moment. But thank you for your time.